Hi, everyone. My name is Cuiping. I am a research manager of the Genomics Initiative at VA Palo Alto. So your genome, like mine, contains 3 billion DNA base pairs. You may have 3 million DNA variations, and up to 50,000 are private to yourself. And these genetic blueprints define who you are biologically. So that's for one genome. Now we have 460 genomes from the pilot of the Million Veterans Program. And in this scale, we have 48 terabytes of pre-processed data and 26 million of single base pair variation and 5 million longer DNA variations. So the data will grow. We need a robust and scalable analysis platform. We team up with Google Genomics to develop and together develop solutions to analyze this large-scale genomic data on Google Cloud. So the Google Cloud platform comprises of 3 components: storage, compute, and BigQuery. BigQuery is a relational database that enables super-fast SQL-like queries. So our solutions, and also via Genomics API, users can boot up MapReduce cluster and run data science scripts in various languages. And you can also explore your data interactively using the BigQuery UI, Genome Browser, and RStudio. And our solutions are comprised of 3 parts. 1, data transformation to compress information. Second, is to develop high-quality QC matches for deriving good quality samples and variants. And third part is the BigQuery queries to mine for biological and medical information. So in data transformation, we adopted the GVCF format. The consecutive no-change positions were compressed. And in this format, we can reduce the file size by 150 times. And the DNA calls were represented in BigQuery table in nested schema. And in this schema, many parameters for variant calling and read alignment can be kept for each sample. And this is unlike the other merge table generated by other tools. And the BigQuery table enables researchers to check for very detailed information in a single table. We developed multi-level quality controls, both at the sample level and the variant level. We consider both principles for individual genomics and population genomics. For example, in this TI-TV ratio by 100,000 window base pair window in a single genome can help us to detect any genomic regions that are error-prone. And this TI-TV ratio considers sequencing depth distribution over the entire cohort, and that can help us to define or detect any variants that are identified with too low or too high coverage. Our data mining solutions enable interactive queries for deriving statistical, biological, and medical information. So we can combine analysis with other cohort. For example, we compare the allele frequency distribution in our cohort versus the 1,000 Genomes project. Our cohort is shown in green here. So as you see that with the decrease in allele frequency, we contributed more and more significantly. So we were able to detect many rare variants because our genomes were deeply sequenced. And we can also try to identify medical information that are actionable. So this is the example of the warfarin dosage effect. Warfarin is a blood-thinning drug. Its excessive dosing can lead to very dangerous bleeding. And by jointly querying our DNA variation with the pharmacogenomics database from GKB, we identified actually 150 subjects, that's a third of our cohort, carry DNA variations that would require a lower dosage of warfarin. And we also identified that 21 subjects carry DNA variations that will require an even lower dosage, extremely low dosage of warfarin. So lastly, to share with you the performance of the Google Cloud platform. So presented here are a few common queries for deriving cohort statistics. We compared it in 5 genomes versus the entire cohort. And in either case, all the queries can be completed within 16 seconds. And for the cost, each query costs less than 40 cents, even for the large cohort. So we think that the solutions that we're building here for analyzing large-scale genomic data is robust, interactive, scalable, and affordable. So here is the team contributing to this work. Our poster is at 1W, so I'll be happy to discuss with you in more details. Thank you.